Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello students, welcome to the TV learning programs where you're going to be learning and watching. I'm teacher Herbert as usual. He's going to be taking you through the biology lesson which is concerning about chromosomes and nucleic acid, which is a unit 3 of senior 5. So, students, I will come to that lesson of today concerning about chromosomes and nucleic acid. Last time we tried to talk about the DNA structure. We talked about DNA replication. We talked about some of the enzymes that are involved in the process of DNA replication. Where I talked about DNA replication is concerning about how the DNA replicates itself and make a copy of its original copy of itself. So today, the lesson is also concerning about chromosomes and nucleic acid. So when you talk about chromosomes and nucleic acid, the learning objectives of today, of that lesson, learning objectives. We're going to be having what you call chromosome to explain what is chromosomes, explain chromosomes. That is number one. Number two, we have to talk about nucleic acids. And the last one, a nucleic acid, where we have to talk about the nucleotides. First, let me just ask you, what do you understand by chromosome? There are a number of definitions of how you can explain what is a chromosome. And we normally say, most of the time, the chromosomes are normally found in a cell. You might say a cell contains the nucleus, which controls and regulates all activities of the cell, including what we call hereditary. That is passing one character or one trait from the parents to the offsprings. Here I can start saying what is a cell. I can say a cell contains nucleus, contains the nucleus, which controls and regulates all the activities of the cell, and heredity. That is to say, passing a character from the parents to the offspring. You can also say that the nucleus is also able to do this because it contains the structure which is called, why is the nucleus is able to do that function of controlling and regulating the activities of the cell. It does that activity, that function of controlling and regulating all activities of the cell because it contains chromosomes. That's why the cell is able to control this. It is because it contains chromosomes. Now, students, let's go quickly to understand what are chromosomes. What are chromosomes? We can say chromosomes are cell-like structures that are found in the nucleus, and they are short and condensed. All right? They must be containing the genes. I can say we have a cell. Inside the cell we have chromosomes. Inside the chromosome we have DNA. On the DNA we have what we call genes. Let's say we have a cell. Inside the cell we have the nucleus. All right? Then in the nucleus, we have chromosomes. Then chromosome contains what you call a DNA. Okay? The DNA contains the genes, a unit of inheritance. So it means that that's how, like I said, a cell, by definition I said a cell is the best functional unit of a living organism. And you can say a cell contains the nucleus. I said the nucleus controls and regulates all activities of the cell. Chromosomes. Now we're going to talk about what are chromosomes. Chromosomes, I mentioned that chromosomes are cell-like structures in the nucleus of plants and animal cells. You can take a note about that. Chromosomes, what do you understand by chromosomes? On your common understanding, what are chromosomes? You can say chromosomes are sled-like structure, sled-like structures in the nucleus of plants and animal cells. So the animal cells contain the chromosome. You can say chromosomes are found in the nucleus of plants and animal cells. And I can say the cell-like structures, which are called chromosomes, they coil up and tightly packed together. They become thickened and shortened and become more compact chromosomes just before cell division. I remind you, we talked about types of cell division. Talked about mitosis, talked about meiosis. And we said both of them, they share the same phases. Where we talked about the interphase stage, we talked about the prophase stage, we talked about the metaphase stage, and we talked about the anaphase stage, and the last one is called the telophase stage. And we said in interphase stage, the chromosomes are not we can say in the interphase stage, we call it the resting stage because there's no cell division there that is taking place there. But when it comes to the prophase stage, we normally say the chromosomes are shortened and condensed and visible on using electronic microscope. So here on a prophase stage, which is the second stage, we can call it to be the first stage because prophase stage is the longest phase among the phases of cell division. All right? So in that prophase stage, that's when the chromosomes are visible and condensed and become shortened and thick and able to become visible on an electronic microscope. All right? So you can also say the cell-like chromosomes coil up and tightly to form a thicker, shorter, and more compact chromosome just before cell division. You can also write that definition of chromosomes. You can call it be chromosomes are made of wrong aggregates of genes. If I say genes formed from a condensed chromatin. So I say condensed chromatin. Once the chromosomes they become shortened and become to be thick, they are able to become visible on light microscope. So I can say chromatins are made up of DNA. If I say what are chromatins now? What are chromatins? Chromatins are made up of the DNA. We talk about the proteins. And most of the time when I talk about proteins, what you see, I myself, what I inherited from my parents, I inherited, as what you're seeing me, you're seeing a protein, because a protein is a building block of the body. So when you talk about chromatins, they are made up of DNA, they are made up of proteins, and also they are made up of RNA, and other biological, and other micromolecules, and other macromolecules. If I say macromolecules, these are big molecules that makes up our body, micromolecules, all right? And all these things are normally located where? They are located in the nucleus, all right? Let me say the chromosome, let me try to draw a structure of a chromosome, the structure of a chromosome. Structure of a chromosome. All right, let's have the structure of the chromosome. When you have the structure of the chromosome, we try to see how the chromosome looks, and we try to see what are the major constituents. You can say this is the structure of a chromosome. The structure of a chromosome. I said chromosomes are cell-like structures, and they are found in the nucleus when it is condensed. All right. So let me try to draw the structure of a chromosome. Most of the chromosome has 2 strands, and those 2 strands are referred to as chromatids. Okay? And they join together. There's a joining center, which is called a centromere. So the structure of a chromosome is like that. This is the structure of a chromosome. This is where the point of attachment, where 2 sister chromatids meet. That one is referred to as a centromere. So you can see this one is called centromere. And this is a point of attachment where 2 strands, where the 2 strands of chromatids meet. You can say these are called chromatids. Chromatids. All right. So the chromosome is made up of 2 strands, and those 2 strands are called chromatids. All right. I said now this is a point where 2 sister chromatids meet. Where they meet, where 2 sister chromatids meet, it is called a centromere. So it acts as a point of attachment where 2 sister chromatids meet. So we are through with the chromosome. Now let's talk about what do you understand by nucleic acids? Nucleic acids. All right. Let's talk about what are nucleic acids. I think everybody comes to know our body is made up of very many biological molecules. Those biological molecules, as I told you, we have carbohydrates, biological molecules. We talk about carbohydrates. We call it the big 4. Our body is made up of carbohydrates, which are energy-giving foods. We have proteins, body-building foods. We have lipids, which is also an energy-giving food. But we normally say the lipids gives more energy than carbohydrates. The reason it requires more oxygen to be oxidized, all right? Compared to the carbohydrates. Carbohydrates gives energy, but it requires less oxygen to be oxidized. So among of the 4 big 4 biomolecules, we talk also about nucleic acid, which is also a building block of our body. So what are nucleic acids? Nucleic acids. I think you're not, everybody is not, this is not something new, because I mentioned the building blocks of our body. We have the carbohydrates, we have the proteins, we have the lipids, as well as the nucleic acids. All right. So let's talk about nucleic acids. When I talk about nucleic acid, I will try to draw something. I say nucleic acids, we call it to be nucleic acid because most of them, they have a functional group of acids. As you well know in chemistry, we have strong acids and weak acids. So these are nucleic acids. These are building blocks of our body. So nucleic acid is made up of 2 things I'm going to mention. It is made up of the DNA and the RNA. Where do we find DNA? I said DNA is found in chromosomes, as well as the RNA is found in there. It is not found, the DNA is found in the nucleus of every living organism. I say precisely I'm talking about the eukaryotes, all right? And then RNA is not found in the nucleus. Instead, it is found in the cytoplasm, all right? And this one plays a big role as far as protein synthesis is concerned, all right? This one, I said DNA. I want to remind you what are DNA. DNA, we call it to be a storehouse of genetic information of every living organism. So what in most of the time, the storehouse of genetic information is the DNA, and also participates in the process of protein synthesis. As you know, I can try to say, we can say we have DNA. From the DNA to RNA, then from RNA, we go to proteins, because our body has to synthesize proteins, all right? And we say proteins are building blocks of our body. So we say DNA plays a big role in protein, in synthesis of protein. We call it, we have that unit which is called protein synthesis, where the RNA does a process of producing what you call messenger RNA and does what you call copying this one strand of DNA, and which is going to be making what you call amino acids in our body. So let's talk about these two things. What are DNA? I think this is not something new to you. When I say DNA, I mean it is deoxyribonucleic acid, all right? And I remember I informed you that DNA is a double-stranded structure. If I say a double-stranded structure, it is made up of two helices, all right? Let me try to show you. This is the structure of DNA. It is made up of two strands. There's one strand and there's another strand, all right? And what is making this, what is supposed to be in the middle of the skeleton? I can say this, call it to be a skeleton. What is in the middle, we have what you call nitrogenous base pairing. And these nitrogenous base pairing, they must be complementary to one another. I think I can remind you, if I say complementary to one another, you must be know that the adenine with the guanine, as well as the cytosine, the thymine with the cytosine. They must be complementary base pairing. Complementary base pairing, it means they have to follow the rule of what you call the Chagaf's rule. The percentage of adenine is equal to the percentage of guanine in the DNA. The percentage of cytosine is also equivalent to the percentage of the cytosine is equal to the percentage of the thymine. So let's talk about what are deoxyribonucleic acid. I said deoxyribonucleic acid, it is a storehouse for genetic information in the cell. Let's have that definition. Can say DNA is a storehouse for genetic information in the cell. Can say DNA is a storehouse for genetic information in the cell. That's good. In the cell, all right? So every cell must be having DNA, and DNA is going to be acting as a storehouse for genetic information. Or you can call it to be, it is a complete set of an organism DNA. A complete set of an organism DNA, it is called a genome, all right? Take that definition. A complete set of an organism DNA is called a genome. If I say a genome, it means it is a genome. I can say it's supposed to be containing the genes, and the genes are the segment of DNA which encodes for a particular trait. If I say it encodes for a particular trait, so meaning what we normally inherit from our parents is supposed to be the genes. Okay? So if I say a gene, it is a unit of inheritance. Or you can call it to be a segment of DNA that encodes for a particular trait. That one is referred to as a gene. You can also say chromosomes are structures that hold these genes. I told you in the DNA, if I say this is a structure, this is the segment of DNA. A point where genes are located, it is called a locus, all right? So we can say a gene, it is, I can refer to as a segment of DNA or chromosome on a chromosome that encodes for a particular trait. If I say a trait, in biology it is referred to as a character, okay? Characteristics, all right? And some of us, we normally inherit very many characteristics from our parents. These characteristics involve the skin color. It involves what you call tallness, shortness, skin color, and etc. All right. Let me talk about what are the major constituents of chromosomes. All right. constituents of chromosomes. What are the major constituents of chromosome? Chromosome is made up of nucleotides. It is made up of histone proteins. If I say histone protein, these histone proteins are normally warped up by the DNA. They normally warped up. They are normally surrounded by. We also have another one which is called non-histone proteins. Non-histone proteins. So these are the major constituents of a chromosome. Chromosome is made up of nucleotides, histone proteins, and non-histone proteins. All right? Let's start with what we call nucleotides. What are nucleotides now? If I say nucleotides, they refer to the monomers that makes up the DNA and RNA. All right? Take that definition. What are nucleotides? Students, think about it and you tell me what are nucleotides. I said nucleotides are monomers that makes up the DNA and RNA. This is the definition. Can say nucleotides, take the definition, take a note of the definition. Nucleotides refers to the monomers. If I say monomers, the monomers that makes up deoxyribonucleic acid. If I say deoxyribonucleic acid, I mean DNA, which is DNA, and RNA, which is referred to as ribonucleic acid. Ribonucleic acid, which is RNA, and these are called nucleotides. And nucleotide has three major components. There are three major components of nucleotides. Students, I'm going to mention these three major components. I can say the nucleotides is made up of what you call a pentose sugar. Let me start with a phosphate group. One is a phosphate group. The second is supposed to be the nitrogenous. It is pentose sugar. A pentose sugar, if I say pentose sugar, it means it is a 5-carbon sugar. A pentose sugar, and the last one is a nitrogenous bases. Nitrogenous bases. So, nitrogenous bases. Here, when I talk about phosphate group, it can just be can just be an acid. If I'm trying to write it, the structure of a phosphate group or phosphoric acid, which is very clear. Let's go quickly to the diagram. I have the structures of deoxynucleic acid and ribonucleic acids. All right? You see the structure. It is a 5-carbon sugar. Here we have the first one which is supposed to be a phosphate group. This one is a phosphate group which is attached to a carbon which is supposed to be number 5. We normally, when you're doing nomenclature, we start what you call clockwise. This is supposed to be, we start from here. This is 1, 2, 3, 4, 5. So the phosphate group is normally attached to the carbon number 5. And we can say this is an adenine, which is supposed to be adenine. As you can see, this is a structure of an adenine, which is a nitrogenous base. Examples of nitrogenous bases, we have nitrogenous bases. We have adenine, we have guanine, we have cytosine, we have the thiamine, and we normally say thiamine is normally found in the DNA but absent in RNA, all right? And also we also have what you call uracil, which is absent in RNA. So you can see this is a structure that presents what you call a nucleic acid. This is a structure which is showing this is a structure which is called a ribose, which is supposed to be RNA structure. So we got here the pentose sugars. Pentose sugars, we call it the pentose sugar because it is a 5-carbon sugar. So what is the common difference between a ribose sugar and a deoxyribose sugar? we normally say on this one, on the deoxyribols, is normally found in the DNA. And on carbon number 2, we have hydrogen. There's absence of in case of the ribose sugar, which is supposed to be present in RNA. We have a deoxyribose sugar in the DNA. We have a ribose sugar in RNA. That's why I said this is A and this is B. So in a ribose sugar, we can say there's what you call hydroxyl group in the second carbon atom. All right? So we have 1, 2. So in second carbon atom, we have a hydroxyl group. Better to circle that hydroxyl group and hydrogen, which is found in the deoxyribose sugar. So that is the common difference that we have from there. So these are examples of what you call these are examples of purines. We normally find in nitrogenous bases, we have what you call purines and pyrimidines. Purines are double-structured, double-ringed structure. But for the case of pyrimidines, for them, they are single-ringed structure. So this example of a parental compound of purine, this is a double-stranded structure. And when you're trying to label them, we normally try to see this is an adenine, and adenine must be complementary base pairing with the guanine. So it means that it is G with A. You can say G with A and T with C, complementary base pairing. And that one follows the rule of what you call the Chagaff's rule. So let us go quickly to the purines and pyrimidine diagrams. As I'm trying to see, we have this one is a double-ringed structure, which is numbered 9 structure, which is called a purine. You can start from here when we are doing a nomenclature of these compounds. This is 1, this is 2, 3, 4, 5, 6, 7, 8, 9. So it means when you are labeling, you do what you call anti-clockwise. Clockwise, not clockwise. You normally go in this direction. Then this one is also a double-ringed structure, which is this is adenine, this is guanine. This must be complementary base pairing. So we have pyrimidines, which are supposed to be a single-ringed structure, which are biomolecules that makes up our body. So we have a single-ringed structure. When you are labeling them, you do what you call clockwise, not anti-clockwise. This one goes anti-clockwise. This one are going what you call clockwise. And when you are going to name them, you consider where on carbon atoms. That's supposed to be nitrogen. We have nitrogen and carbon. This is 1, 2, 3, 4, 5, 6. So pyrimidines have 6 numbered carbon atoms, but for the case of the purines, they are numbered. I can say they are numbered 9, double-structured and numbered 9. But these one are numbered 6. So this is an example of cytosine. So purine is made up of the cytosine, uracil, and thiamine. All right? And I said this uracil is found in RNA, but absent in DNA. All right? So simply, let me just give you a summary of it. If I'm trying to give you a summary of it, you can say, a summary of that, you can say nucleic acids are normally made up of DNA and RNA. And they say in the nucleic acid, we have what we call chemical structures. These chemical structures are called nucleotides. And nucleotides are made up of 3 major compounds. It is made up of a phosphate group. And the phosphate group must be acting as a backbone or the skeleton of the DNA, all right, which provides its protection, all right? We have a pentose sugar. A pentose sugar, as I mentioned it, this is a 5-carbon sugar. This one is a 5-carbon sugar, so it is also made up of a pentose sugar. And without forgetting that there is also what you call nitrogenous base. The pentose sugar is made up of 2 things. It is made up of deoxyribose sugar and a ribose sugar. I think you are following me. The pentose sugar is made up of deoxyribose sugar and a ribose sugar. Then nitrogenous base is made up of what? It is made up of purines and pyrimidines. Then purines are made up of what? Student, what are the major constituents of purines? Purine is made up of adenine and guanine. And then pyrimidines are made up of cytosine, thiamine, and uracil. Thank you for being with me in my lesson. See you next time.